Hello, grade 11 students. In this video, we're going to talk about the mutations worksheet solution. So let's start with question number one is talking about the Dutchen myopathy. The Dutchen myopathy is a genetic disease due to a single gene. This gene, situated on chromosome X, consists of more than 2 million pairs of nucleotides. It permits the synthesis of a muscular protein which is called the dystrophin. The fragment of the transcribed strand, DNA represented below, corresponds to the amino acids between 109 and 114. So this means that this is a portion and not the complete gene of dystrophin. So the fragment of they mentioned here, the transcribed, the template, meaning that it's the non-coding. Transcribed, template, non-coding. Okay, so this sequence is the transcribed. So the mRNA is obtained by obtaining the complementary strand. So you have to make it complementary. So C, the complementary is G. C, the complementary is G. A, the complementary is T. But in the mRNA, we don't have T. We have to put U. So it becomes GGU. A, also it's U, since there is no T. A, also it's U. C, it's G. T, it's A. A, it's U, the complementary U and U. C, it's a G. C, it's a G. T, it's A. A, it's T, so we have to put U. T, it's A. It's U. And finally, T, it's A. Following several mutations, the sequence of the nucleotides of the genes, they can change. Dutchen myopathy, or what's called Dutchen muscular dystrophy, is due to one of these changes. Part A. By the help of the genetic code table, indicate which of the amino acids between 109 and 114 is responsible for the dystrophin. Number 2. Determine the consequences of a mutation of the polypeptide sequence taken of the fragment of the transcribed strand of the DNA. Here they mention here the number of the nucleotides. So number 1 for the nucleotide. Here it's 2, 3, 4, 5, 6, 7, 8, 9, and this is nucleotide number 10, 8, 11, 12, and so on. So let's with first part. Let's write the complementary strand since this is the transcribed. So this is the transcribed. So it's the non-coding. So what I have to do is to make it complementary. Okay? So the complementary strand. So it's CCA. Let's write it down. CCA. This is the first three. CCA. Then we have AAC, TAA, ACC, TTA, and TAT. So first CCA, AAC, TAA, ACC, TTA, and TAT. Let's make the complementary. So the complementary for the mRNA. So here, mRNA. Okay, complementary, but we don't have T. Instead of T, we have to replace it by U. So the complementary of C, it's G. Complementary of C, also G. A, it's U. A, it's T, so no T, U, U, G. T, it's A, and A, it's T, T. No T, what I have to put? I have to put U. So you have to pay attention to this. Okay, so this is these mistakes. Always you fall in these mistakes. So you have to put instead of A, you have to put U and U. Okay, pay attention to this. Then let's move now to the A. It's going to be U. Okay, don't fall in mistake again. C, it's G. C, it's G. T, it's A, U, and A, U, A. Here is the sequence of the mRNA. So this is the sequence of the mRNA. How can we determine the sequence of the amino acid? Simply using the genetic code table. So using the genetic code table, GU. So the first letter is G and the second letter is G. Let's move here. This is the second letter is G. So it's glycine, GLY, GGU. Okay, UUG. Do you notice here four codons? They code for the same amino acid. This is called redundancy. Where more than one codon, they code for the same amino acid. UUG. So the first letter is U and the second letter is U. UUG. It's leucine. LEU. Okay, and let's continue. For AUU. AUU. Where's AUU? A, first letter is A, and the second letter is U, and the third letter is also U. So AUU, it's isoleucine, ILE. Let's move now. UGG, the first letter is U, and the second letter is G, and the third letter is G, so it's tryptophan, TRP. Then AAU, first letter is A, second letter is A, and the third is U, so it's asparagine, ASN. And finally, the last nucleotide, AUA. So AUAA, first letter, second letter U, AUA is also isoleucine, ILE. Here we determined the sequence of the amino acid. So the indicated sequence is that of the transcribed strand. So the sequence of the mRNA is obtained by the complementary. So this is the sequence of the mRNA. According to genetic code table, we determine the sequence of the amino acid. It's glycine, leucine, isoleucine, tryptophan, asparagine, and isoleucine again. Part B. They said if T replaces C at the position number 12, the nucleotide UGG become UGA. So UGA, this is a stop codon. So there is stop codon. So the synthesis of dystrophin will stop after the amino acid 111. Also, if T replaces C at position 6, the 110 codon UUG become UUA. So according to this genetic code table, these two codons, UUG and UUA, let's say UUG, leucine, and UUA, UUA, also leucine. So both of them, they code for leucine. So the dystrophin will not be changed or modified. So this is a kind of silent mutation. Now, let's move to question number 2. Question number 2 is talking about the oxytocin and vasopressin. The oxytocin and the vasopressin are two hormones. They are synthesized by the post-pituitary glands of the mammals. The first has the uterus as a target organ, and the second has the arteries and the kidneys. The document below is showing in number 1 the sequence of the DNA bases coding for the oxytocin, and in the second one coding for vasopressin. Pay attention here, coding. Okay, so they said coding. So if you want to determine the sequence, pay attention here. How can we determine the sequence of the mRNA, and how can we determine the sequence of the amino acid? They said the coding. So it's non-transcribed, non-template, it's the coding. So the mRNA is similar to the sequence, but instead of T, it's replaced by U. Part A. Determine using the genetic code table the sequence of the amino acids of these two hormones, and then we have to compare the number of differences between the two DNA strands to that the chain in the polypeptides, and also explain why the differences between the sequences of the nucleotides do not necessarily cause a difference in the sequence of amino acid. A few minutes before, we answered the part C, if you remember. Part A. Let's start with the oxytocin. So I want to determine the sequence for the mRNA sequence. Okay? mRNA. As I said, this is the coding. So it's the same. Instead of T, it's replaced by U. So TGC instead of T, UGC. Then UAC. Then AUC. The same. Instead of T is replaced by U only. Since this is the coding. AAC. And then we have TGC, it becomes UGC. Okay? And CCC. CUG and GGC. Pay attention here. What did we change in the sequence? We changed only this. T is replaced by U. Also T is replaced by U. T is replaced by U. And here the T is replaced by U. And here also the T is replaced by U. Here. Okay? So here are the changes in the sequences. By this way, we can determine the sequence of RNA. So the sequence of the oxytocin will be UGC, UAC, as we determined their sequence. They will code according to the genetic code table. Okay? So we find them: cysteine, tyrosine, isoleucine, GLN, asparagine, cysteine, proline, leucine, and glycine. The second sequence. Let's move to the second sequence, which is for the vasopressin. Also, the same sequence, mRNA, the same sequence, instead of T is replaced by U. So it becomes UGC, UAC, UUC, CAG. We're working with this sequence, okay? So pay attention. CAG, AAC, UGC, CCA, AGA, and finally GGA. Let's note here what's the difference between them. Here T is replaced by U, and here T is replaced by U. These two T's, they are replaced by U, and then we have this T is replaced by U. Okay, so this sequence will code for the sequence of the vasopressins, the amino acid sequence. But wait a minute, if I want to compare the sequences of the DNA, okay, first of all, TGC is TGC. It's the same for both vasopressin and the oxytocin. Second, TAC is TAC. It's the same. Let's move for the comparison. Third one, ATC. Here it's TTC. So the first change is here. And the nucleotide number 7. Okay? And then CAG is the same. AAC is the same. TGC here. And here we have TGC. Okay? CCC here, it's CCA. So the A is replaced by, C is replaced by A. CTG, here it's CTG. Here we have C is replaced by A. TG and G and A. GGC, here it's GGA. So here also there is a difference. Let's move to the question. So the amino acid sequence, it's here. According also to the genetic code. So here are the two sequences. We determine the differences between them, these two sequences. So here TGC, TGC the same, TAC, TAC. Here the first difference, A and T. CAG, CAG, AGC, AGC, TGC, TGC. Here we have A is replaced by C here. CTG, all of it is different. CTG here is we have AGA. GGC is replaced by GCA, so C is replaced by A here. You can determine this by numbering them. So here it's position number 7. 3, 6, 7. Here we have 12, 15, 18, 19, 20 here at position number 21, at position number 22, 23, 24, 25, 26, and at position 27. So let's determine the user genetic code table. We did this part. Compare the differences. We compared it orally. and let's explain why the differences between the sequences of nucleotide doesn't necessarily cause any difference in the amino acids. So there are six differences. The position suffix, as I said, 21, 22, 23, 24, and 27. And only two differences in each polypeptides at the position 3 and 8 in the amino acid sequence. So six mutations or six differences are not necessarily expressed in the structure of the polypeptides because the genetic code table is redundant. What the meaning of this? It means that more than one codon they can give the same amino acid. That's why some mutations can change the codons into equivalent codons that can code for the same amino acids. This is called silent mutation. Okay, thank you. Bye bye.